Hello, everyone. This is Kelly Ebersole, the Executive Director of the International Wheat Genome Sequencing Consortium. And I'm joined today by Sean Walkowiak from the Canadian Grain Commission. And he's going to give us a webinar today as part of the IWGSC Webinar series. So first, I'd just like to give a quick overview of the International Wheat Genome Sequencing Consortium. We have about 2,800 members. We have 9 sponsors, over 800 institutes and companies who are involved, and we have members and sponsors and institutes in 70 countries. The webinar series itself is supported and made possible by the generous sponsors for the consortium. So we'd like to thank Arbor Biosciences, BASF, Florimond Desprez, INRA, Lima Grain, Kansas Wheat, RHET, Syngenta, and the University of Adelaide for their financial support. The IWGSC is in its phase 2, or working towards our phase 2 vision. And the goal is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. Our activities include expanding the IWGSC Arbor Biosciences collaboration to include more opportunities and resources for scientists, to publish the IWGSC RefSec version 2.0 that will include gap closure of version 1. Also increasing our RefSec annotation, including both manual and functional annotation, and then annotating versions 2.0 and 2.1, what we think will be 2.1. We are still trying to launch our Wheat Diversity Project to get reference sequences of at least 8 land races that represent the breadth of wheat diversity. And then, of course, continuing to try to provide pre-publication releases of genome sequences for elite wheat cultivars and other genomic resources. And then the last thing that we are doing this year is our IWGSC webinar series. Just a quick reminder that our webinar, our next webinar will be with Cristobal Uau from John Ennis, and he will be talking about a roadmap for gene functional characterization in wheat. Just as a quick reminder, the webinar is being recorded and will be posted on the IWGSC YouTube channel. We will be posting it at a later stage than normal because for this particular webinar, we're getting a peek at what has not yet been published. And so as soon as the manuscript has been published, we'll be able to release the channel plus the PDFs of the presentation. You can subscribe to the channel to never miss a new upload, so you could get an immediate notification. You can do that by going to this link. And the presentation will be followed by a Q&A, and so please submit your questions in the Q&A panel. Do not use the chat, as I won't be looking at that. And you can use the chat to talk to fellow attendees or to the organizers. Also, you can download at least my slides, which will have these links from the handout panel. So with that, oops, sorry, went the wrong way. I'd like to turn it over to Sean, who will talk with us about the 10 Genome Project. So Sean, thank you for agreeing to present today, and we look forward to your presentation.
Thank you, Kelly. Yeah, so thank you everyone for coming on to the webinar today. I'm really excited to be presenting on behalf of the 10+ Genomes Project. Curtis Posniak was originally scheduled to present, and he sends his regrets. He had another meeting that he could not get out of. So I'm presenting on his behalf today. And today the presentation is going to focus specifically on alien introgressions that we were able to identify in some of the 10 plus wheat genomes lines. So to sort of backtrack a bit and to sort of discuss how we've gotten to this point. We know that the IWGSC RefSeq sequence was published in 2018, and this is really a monumental publication. And it's, and I just looked at the Google stats today. The manuscript already has 539 citations in the short period that it's been published, which is really exciting. And the publications actually, the citations span not just wheat, but all sorts of crops, including cotton, blueberries. So it's been really impactful to both wheat as well as other crops. But one of the challenges that we've been observing is that, especially in wheat breeding programs where we're looking at trait improvement, is that a single genome does not capture all of the variation that's important for improving wheat. So shown on the left here, we have a Venn diagram that sort of represents the diversity that we might see in different wheat lines. So you'll see that there's some regions of a genome that might be unique, that's only present in one line. There might be some regions of the genome that are shared across 2 or more lines, and then there'll be some regions of the genome that are shared amongst all lines. And it's this variable regions within the genome, the unique fraction, as well as what we call the dispensable fraction that's only shared amongst a subset of lines. It's really important for giving rise to trait variation. So for example, increased yield or disease resistance. And this is the regions that we really want to get at as breeders and research scientists, because this is where the genes are that are important for improving wheat. So genome sequences are valuable because they give us a complete atlas of the gene content, and they can really help support both gene discovery and marker development. And by sequencing additional genomes, we get more information about structural variation between individuals, copy number variation of genes, presence-absence variation, as well as single nucleotide variation, or haplotypes that could be giving rise to this trait variation. So the idea here is by capturing information from multiple genomes, we can really get at that variation and then start to improve breeding, speed and accuracy, and then we can more rapidly respond to producer needs, and we can then really tackle those top priority traits. So the idea of the sequencing approach is that it should be hierarchical. And what this means is that just like RefSeq has been a useful reference for the wheat scientists to study their genes, and that's because it has a really complete genome sequence at the chromosome level. However, this isn't really cost effective to do for all wheat lines. But if you have a good set of references, then you could really get at most of the variation that you're likely interested in. And so the idea is to have sort of a tiered approach where you have multiple high quality genomes. And then if those genomes contain the variation that's important to you, you can actually sequence your lines at a reduced level of depth and be able to still capture that information from those high quality reference genomes using them as a guide. And likewise, if you have populations, so for example, individuals from a biparental cross, you might have hundreds or even thousands of individuals, so it might be unrealistic to even sequence those at medium depth, but you can impute their genomes from these high quality genomes using GBS or low depth sequencing or exome capture, or even 90K array or 820K array, for example. So the 10 Genomes Project was set up to really capture variation from different breeding programs so that we'll have more references available so that we could do these other studies on lines within breeding programs. So as part of the 10 Wheat Genomes Project, we've sequenced 2 Canadian spring wheat lines, CDC Landmark and CDC Stanley, which were done by Curtis Posniak and Andy Sharp. We have a US winter wheat line, Jagger, by Jesse Poland and Gary Mulbauer. We have a German winter wheat line, Julius, which was produced by Nils Stein and Klaus Meyer. Winter wheat line, Arena LR4 from Switzerland by Beat Keller. Two Australian varieties, Mace and Longreach Lancer by Peter Langridge and Kenneth Chalmers, I believe, and a Japanese variety, Norn 61, French variety, SY Mattis from Syngenta, as well as one spelt wheat, which was done by Asif Distapel. And to complement these chromosome level assemblies that were sequenced using the Energene pipeline, we've also constructed 5 scaffolded assemblies for Cadenza, Paragon, Robigus Clare, which are UK lines, as well as Weeble, which is a CIMMET line. And these were done by folks at the Earlham Institute, John Ennis, the Natural History Museum, and NIAP. So to give you an idea of where these genomes land in terms of hexaploid wheat diversity, what we did is we extracted the data from 2 recent studies where they did exome capture sequencing of multiple lines of hexaploid wheat. So those are illustrated here in this PCA plot as gray markers. We also included 16 lines from the Dawn project, which are the orange markers. And then here with the black markers, and they're all individually labeled, are the 10 wheat genomes lines. And you can see that they span quite a good diversity of the hexaploid wheat. And we see grouping that makes sense based on breeding history and genome diversity. So we have the Asian lines grouping together Australians, the European winter wheats, UK springs, North American and CIMMET lines largely grouped together. Jaeger is a little bit off, and it goes closer to some of these winter wheats because it's a winter type. And then spelt is sort of doing its own thing over here. So the approach that we used to do the sequencing and assembly, we had paired end and mate paired libraries with different insertion size. We used 10x genomics and a high C. So this is quite similar to what was used for RefSeq. We used the de novo magic pipeline or the RAP2 assembler. Then we manually inspected different scaffolds and contigs, and we did error correction to break any chimeras. We then ordered and oriented the scaffolds into the pseudomolecules to represent the chromosomes, and using the high C, we can do corrections for those. For each of the lines, we also did chromosome squashes, and we manually inspected each of the chromosomes to be able to detect some interesting things that we'll get to in a bit. And for one of the genomes, CDC landmark, we also did whole genome Oxford nanopore sequencing, and we were able to support the scaffold arrangement in the pseudomolecules using that technology. So here's a sort of a snapshot of the comparison between all of the genomes. So this is an all by all genome comparison, and it's sort of sorted by chromosome. And what we see here is we're mostly getting these diagonal lines, and that indicates that the genomes are highly syntenic. So they're aligning in a sort of linear fashion between each genome in sort of a one-to-one fashion. But we do see some things that are a bit neato. So for example, if we look at this red box here, this is Longreach Lancer, and there's something peculiar happening here on chromosome 2B. It looks like it's not aligning well to anything else. And on chromosome 5B and 7B, we have something wonky going on with Arena LR4 and SY Mattis. But if you look at their 5B and 7B chromosomes, they align quite nicely. So they're the same, but all the other lines are different. So this is sort of a nice quick snapshot to show that most of the genomes are highly collinear. So the structure of the genomes are fairly similar, but we do see some interesting things here that I'll get to later on. So here are some of the assembly statistics. So Chinese Spring is up here. And you can see that for all of the newly assembled genomes, we're getting a roughly similar genome size. This is the assembly length is what was able to be included into the pseudomolecules or the chromosomes. And NR genes pipeline, especially including the the 10x genomics data, has significantly improved over time, and we're seeing much better N50s. So we're seeing, on the order of almost 50 megabases if you look down at the bottom there for SPELT. So these are really good, high-quality genome assemblies. So in addition to generating the sequences themselves, we also work towards getting some annotations on these. And for our initial approach involves using the Chinese Spring gene models, since a lot of effort was placed into getting those done well. We chose those models and then projected them onto each of our newly sequenced genomes. And then we looked at to make sure that they're in the right order and orientation in the genomes, to look at if there's syntenic orthologs. So we make a grid of these, and then we can filter for the best high matching scores, and then as well as orphan genes that are going to different locations to generate some gene annotations. So again, the idea is that we take the Chinese Spring here on the left, and then we project those genes onto the new genome. And then what we can do is start comparing the genes between each of the genomes. And what we see is over 80,000 genes are what we call complete, and this means that they are present as one copy in each genome. And then there are another smaller set of genes which are present in 2 to 10 lines. So this means it's missing in one. And then there's a smaller set of genes that are have something more complicated going on, where there's multiple copies in a single genome, or there's missing in more than one line. And what we can do is we can start to cluster the patterns of presence-absence variation, for example. And what we see is, again, the Asian lines grouped together, shown in dark green on the top left corner here. Then we got the winter wheat sort of making their own little cluster. Long-reach Lancer is doing its own thing a little bit, as is SPELT. This is likely due to that thing I pointed out on chromosome 2b that I'll get to later. And then some of the other North American and Australian lines are largely grouping together. In terms of gene annotations, we can also look specifically at certain gene families. So this is an example of an analysis using the NLR genes. So these are nod-like receptors. They're typically involved in disease resistance. And we actually see a lot of diversity in some of these gene families. So for example, in a given genome, you might have between 2,000 and 3,000 of these genes. But as we sequentially add more genomes, we're actually finding that these genes are not well shared between each of the genomes. There's only about 31 to 34% of them that are actually shared. And as we start to add genomes, we're getting more and more unique NLRs, up to almost 8,000 NLRs across all the genomes which we sequence, which is quite impressive gene expansion within this gene family, with 22 to 192 being unique in a given genome at this end stage. And some of that, as you can see, decreases as we start to adjust the threshold for identity between them. So this shows that there's, not just gene expansion, but also probably selective pressures within those genes that are causing them to change. And these are important for disease resistance. For example, here there's a picture of some leaf rust. So this is typical for the gene-for-gene interactions, for example, for some of these pathogens. In addition to annotating the gene space, we also annotated the transposable elements. And we get around 500,000 or so transposable elements in each of our genomes, which is similar to RefSeq. And it's interesting that this sort of shows a linear relationship based on genome size. So this gives us good confidence that the wheat genomes that we're generating in this pipeline are complete. And we can, again, similar to the gene space, start to compare the patterns of TEs between the different genomes. And we see similar patterns of clustering. So the Asian lines grouped together, we get some of these North American and Australian lines grouping together, and then you have these winter wheat lines also grouping together when we compare their TE space. So we can again look deeper into different elements or types of elements of TE. So this is an analysis of Angela elements, which are a type of transposable element. And what we could do is we can extract them and compare them between the genomes and even within the genomes. And we actually see some really interesting patterns. So here on the left is a PCA plot showing the alignment of the different Angela elements. And what we see is they tend to form these clusters of different types of Angela elements based on likely their ancestry. And if we look at where they land on the genome, for example, this A3 group is almost exclusively on the A subgenome, whereas A6 is almost exclusively on this B subgenome, and then A4 on the D subgenome. But then you see some really interesting things sort of highlighted by these red arrows. So you have, for example, this region on 2D that has this A3 type, which is typical of the A genome. So this suggests that there's maybe some interactions between the subgenomes, and we have a region here that has something on it that's either from the A genome or from a relative that's closely related to the A genome donor. Likewise, on the A genome, we see some interesting things that don't look like it's from the B or D subgenome. It has this A1 group that looks like it's something else entirely. So this led us to think that maybe these regions are alien introgressions, so sequences that have been derived from other species. So we started to dig into that a little bit further, and this is sort of showing the same data but represented in a slightly different way. So here I'm plotting chromosome 2A, and now we're comparing all of the genomes that we've sequenced. And here on the bottom right, we have the legend. So if it's red, it means that it's unique to a line. If it's blue, it's present in 2. If it's slightly lighter blue, I guess it's present in 3. And if it's green, it's present in 4. So if you look at this region here on chromosome 2A, there are 4 lines, Jaeger, Mace, S.Y. Mattis, and C.D.C. Stanley, that all share the same TE elements for the Angela elements. Whereas, Spelt is sort of doing its own thing. It's sort of an outgroup. It's interesting that these 4 sort of carry the same region here. And if you look at chromosome 2B, Longreach Lancer, so this is that one in the dot plot where there's a massive gap. It has this large struts. It's very dense in these unique transposable elements that are not present in any of the other genomes. And then on chromosome 3D here at the end, we have a dense set of unique transposable elements in Longreach Lancer as well. So I'm going to talk a little bit more in depth about these 3 examples now. So to start with the example on 2A, we know based on pedigree that Jaeger and C.D.C. Stanley carry the VPM1 introgression from Agalops ventricosa. And this is a well-characterized introgression. It's known to carry these rush genes LR37, YR17, and SR38. And it also has recently been shown to give rise to resistance to wheat blast, which is an emerging pathogen of interest because it recently traveled from South America to Bangladesh. So there's a lot of interest in developing resistance to wheat blast, and this region recently has shown that it can provide some resistance to that as well. So here's a picture of the wild grass, and then we did chromosome squashes. We could do labeling to show that the introgression is on 2A. And here's a field. I think we have Jaeger on the left, which is that carries the introgression, and on the right here you can see this yellow divide. And that's because this is, I think, a rust nursery, and you can see the rust that's showing up on those wheat plants. So if we look at what's going on in the genomics here, we have this unique region at the end of at the bottom here in the gray. We have this unique region that's with the green that's corresponding to these 4 genomes. So we were surprised that Mace and S.Y. Mattis are also carriers of this. Here are the projected genes and their density across the chromosome. underneath it in blue. And just underneath that, we have the yellow, and we see this at the very beginning, a really low sequence identity to Chinese Spring, further supporting that this is a different region or an introgression. And then we actually did whole genome sequencing of multiple accessions of Agalops ventricosa, and when we map the reads to the genomes of the carriers, the first little bit here lights up, supporting that this is in fact derived from this wild species. When we do a close-up of the alignment of the region, which is here on the left, and we can align them both to Chinese Spring, which is the left panel, and then Jaeger, which is the right one, we can see that when we align Chinese Spring to Jaeger, for example, it doesn't align well at all. Likewise, CDC Stanley, Mason, SY Mattis don't align to Chinese Spring, whereas most of the other lines seem to align fairly well to Chinese Spring, with the exception of Spelt, which looks like it has some unique sequence within that region. On the other hand, when we align to Jaeger, you can see that CDC Stanley, Mason, SY Mattis align quite well to Jaeger, and all of the other lines do not. So this again supports that all four of these lines carry the same introgression, and that the breakpoint is more or less the same. If you look at the bottom of this graph here, you can see we've labeled the position of megabases, and you notice that the same region that's 25 megabases in Chinese Spring or other non-carriers is actually 33 megabases in the carrier line. So this is actually affecting the chromosome size, and there's some extra sequence in that region, which is could be giving rise to some of this trait variation. So we did GBS sequencing. So this sort of goes back to that pyramid I showed you before. And using the unique tags from GBS, we can quickly sort lines into whether or not they are carriers or carriers based on the presence of tags that are associated with the introgression or not. And when we do this in 3 different wheat panels, we see that the frequency of the introgression has been increasing over time. And then when we look at our phenotyping data of the carriers and non-carriers in different field locations, we can see that there seems to be a positive association with this introgression and yield. So that's an interesting new finding. We can also look more closely at the genes within this region. And it shows some synteny in the gene structure to all the other subgenomes of the same region in the other subgenomes. And again, the region's about 33 megabases. There's about 535 genes. And when we looked, it was quite interesting that over 10% were cytochrome P450s, which are associated with biotic and abiotic stress and have been implicated previously in yield. And over 10% were also had NB-ARC domains or were annotated as NLRs and are likely giving rise to increased disease resistance, which could be giving rise to that, that rust and blast resistance. Then we can do a direct comparison of the genes on 2A and 2N, and these are a tree of the NLRs between them, and you can see that they actually have diversified quite a bit from what's in the other genomes, and there's more of them. So this is an important variation that's likely giving rise to that disease resistance. So I'm going to switch gears a little bit, and I'm going to talk about now that sequence on 3D in Longreach Lancer. So this is also, when we looked at the pedigree of Longreach Lancer, we saw that it has, I think, Lang, and Lang is a carrier of a tall wheatgrass introgression that was developed by Ernie Sears, and it contains SR24 and LR24. And again, this introgression affects the total size of the chromosome, as shown on the top right. So on this plot is similar to the one I showed before, where you have at the end this red bit, which is corresponds to the introgression. You see that there's a drop in projected genes, so this indicates that this region is perhaps a little bit more distant. We did whole genome sequencing of multiple Thynoparum ponticum, or tall wheatgrass accessions, align them to the genome, and it lights up here at the end, letting us know that this is in fact introgression derived from that species. And this sort of goes back to that pyramid. We did 10x genomic sequencing of AAC Concord, which we hypothesized to carry the same introgression. And when we align it to long-reach lancer, the whole chromosome lights up, including the end, suggesting that it is indeed a carrier of the same introgression. And I'm going to sort of go on a bit of a tangent here. So if you recall, with the IWGSC RefSeq manuscript, there was the breeding component, and we focused on this wheat stem sawfly story with a solid versus a hollow stem, where a solid stem gives resistance and a hollow stem gives susceptibility. And we found this region on the end of chromosome 3b that was associated with stem solidness, and we associated it with a gene that had copy number variation. But it turns out that this region on 3b is sort of the same region on 3d that has been replaced as part of this introgression. And when we do some mapping between AAC Concord, which carries the solid stem locus on 3b, with CDC Hughes, which carries the same locus on 3b, we see some differences in the phenotype of solid stem. And this was surprising since both carried that same locus on 3b. And when we did some genotyping, we discovered that there was a massive QTL on chromosome 3d that corresponds to the same region on 3b. So this looks like another locus that's contributing to stem solidness, and it seems to have an additive effect. So in this population, this 3b locus is fixed, but when we add this 3d locus that comes from the introgression, which is right where this QTL stacks up, it's more solid. So there must be something interesting going on here in the genes that are interacting with 3b that are able to give it this additive effect. And this was done in association with Dr. Richard Cuthbert at Agriculture and Agri-Food Canada. And we're currently working on trying to tackle this a little bit further. I apologize for the ugliness of the slide, but essentially we did RNA-seq on a whole bunch of different lines that either don't carry either the 3b or 3d locus, just carry the 3b or just carry the 3d. And we're able to, using RNA-seq, which is a lower tiered sequencing in that pyramid, confirm that some of these lines carry that introgression, that the breakpoint is the same. And now we're looking at the expression of the different genes across that interval, as well as the entire genome, to figure out how mechanistically this solid stem phenotype is working. So now I'm going to shift again to this other example on 2b. So again, this entire chromosome more or less didn't align when we looked at those dot plots. And what we discovered is that this introgression is almost 427 megabases of unique TE patterns on long-reach lancer. And this region doesn't align well to Chinese spring at all. And we sequenced Triticum timofeevi, and we saw that it aligns quite well to this region that has these unique TE elements. And this region, looking back in the pedigree, seems to be associated with SR36. So to sort of summarize what I've shown you thus far on the alien introgressions, sort of going in the reverse order, we had this really large region, almost the entire chromosome, it's massive, that contains SR36, which is likely derived from Triticum timofeevi. We also have this region at the end of chromosome 3D that is derived from Thanoparum ponticum, which we knew contained LR24 and SR24, but I showed you today is also associated with solid stem. Then we have this region at the beginning of 2A, which is from Aegilops ventricosa, this VPM1 introgression, which we knew had some rust loci on it, as well as blast resistance. And I showed you today that it's also associated with yield, and it has a whole bunch of interesting genes, including R genes and P450s, and that there's a lot of diversity in those R genes, including unique ones. So if we go back to this chromosome all by all genome comparison that I showed you previously, we sort of we tackled this guy here on 2B, the one in red, but I didn't get to this 5B, 7B one yet, and I'm gonna sort of get to that now. So what's going on here is that we have a Robertsonian translocation between chromosome 5B and 7B. And in a non-carrier such as Chinese spring, we have the normal chromosomes, but then we have this translocation event that happened here on the long arm of 7B and short arm of 5B. and that results in these chromosomes down here, with this upper one that contains the long arm of 7b, as well as part of the short arm of 5b and the entire long arm of 5b. And this guy is massive. He's 993 megabases long, so longer than your chromosome 3b. So this is now the longest chromosome in these carrier lines. Then we have this tiny guy down here, which will contain the short arm of 5b, part of the long arm of 7b, and then most of the entire long arm of 7b. Shown at the top here is some high-C plots. So these are contact maps and the data for SY Mattis and NORN61, which have been aligned to the Chinese Spring genome. And on the vertical axis, we have 7b, and on the horizontal axis, we have 5b. And what we see is we have these associations between 5b and 7b and SY Mattis. And in NORN61, it's nothing going on, supporting that there's no association between these two in a non-carrier line. And again, all these have been, large cytological events have been supported through chromosome squashes and staining. So here's this really, really massive guy. And then here's that wee guy. And this is, I think, SY Mattis down here. So to help make this data more accessible, we've developed visualization tools so that these genomes can be directly compared, even by non-experts. So shown here is a screenshot that I sort of mashed together of SY Mattis compared to Chinese Spring, and you can, clearly see this translocation event. And you see here the same thing visualized as a dot plot. And these are fully interactive viewers that you could use. So this is a sort of a screen video of the viewer. So let's see if this works here. Yeah. So at the top right, we have these drop-down menus. So here we're selecting Irina Alarfar 7b chromosomes, and we're comparing them to, I think, Chinese Spring. You can see the translocation. On the bottom left, we have the dot plots. You can even adjust the threshold for comparing different sequences between the genomes. You can select individual chromosomes. You can toggle between them. And on the bottom, you can even zoom into different regions of the chromosome. So these are really interactive tools that can be used by any user. You can even customize a lot of the different analyses. So at the top here, there's a dashboard where you can insert your own tracks and you can plot, graphs above each of the chromosomes. So these are very valuable tools that'll be useful for people to be able to compare these genomes. So this is another tool that we developed. It's sort of similar, but instead of having linear plots, it has them in a circular viewer, so something similar to Circos, for those that are familiar with that. And there's also some automated tool that we can sort of see here is rearranging the chromosomes automatically to minimize collisions. You can click and drag different chromosomes. You can select regions. So if you see here on the right, there's some linear views, and this is showing the synteny between those regions. So you can select them individually. It toggles between them. You can invert them, flip them. You can also add your own tracks. So this at the top right, it says load files. You can add your own data. You can plot, histograms, bar graphs, heat maps, whatever you want. So now I'm going to switch gears and show you another viewer, and this one's less designed for synteny, but more to show genome sequence variation. So these are the SNPs, for example, to highlight different haplotypes. Oops, let's see if this video works. Oh, here we go. So this also has a drop-down menu. So here I selected chromosome 2A, and then you click show haplotypes. You can adjust your bin sizes. When you click on any region, it shows you the region that corresponds in the other genomes that are identical. So you can see which haplotypes are shared amongst different lines. So right now I'm highlighting here the 2A introgression that I talked about in the talk today, and those 4 lines carry that. But you can select any chromosome, you can go between each of them, and it shows the different haplotypes that are shared amongst the different assembled genomes. We have lots of different tools. As I mentioned, here's our landing page for our website. This is a landing page for that linear and circular viewer. So if you go to this website, you can click on the drop-down menus. You can select the individual genomes that you want to compare. On the left-hand side, on this blue button here, you can click on the linear plots, or you can click on the circular plots, and it'll generate those automatically for those two genomes. And then you can work with it and upload your data afterwards. We also have a BLAST database shown here on the bottom left. And I showed the haplotype database here. It's currently hosted at this IP address, but we're going to migrate it to this website shortly. And probably more importantly, all of the data for all of the genomes that we presented on today are available for download, including the gene projections and the TE annotations. They're all available on the FTP site at IPK. And many of the people likely on this call already have seed for these lines because they've gotten in touch with us. And we've been really careful to make sure that the seed that we are sending out are directly derived from the lines which have been sequenced. And we're having them redistributed through SeedStore. Several of the lines are already available on SeedStore. Some of them we're finalizing the agreements for that now. But if you have seed or if you want seed, it's going to be available through SeedStore. And this is sort of an ongoing project where we're continuously developing new resources for them. So for each of the lines, we've also developed populations that can help us really get at that gene space and identify causal genes for in-demand traits. So one of the approaches that we're using is mutagenesis. So shown on the top left here is our gamma chamber. It has, I think, radioactive cobalt and EMS, and we sort of dose the seeds with these with different levels of doses. And you can see here the different doses for gamma, for example, and we sort of optimize the dosage, and then we can plant these out and advance the populations. And likewise, we're also developing a NAM population where we've crossed CDC landmark with each of the lines, and we're advancing those as well. And then you're going to have all sorts of different recombination events between the lines that can allow you to help map your traits of interest. And in both populations, the mutant ones as well as the mapping ones through biparental crosses are being phenotyped, they're being sequenced so that we can really develop this massive resource that can allow us to really get at those genes that are in those genomes. So to summarize, the project has thus far constructed 15 genome assemblies, 10 reference quality, and 5 scaffold assemblies. And today I've been able to show you that these cultivated genomes have a lot of interesting diversity. So I showed you an example of that with genes and gene families, and the example I showed was the NLRs. I showed you that with the transposable elements, with the RLC Angela elements. And I also showed you examples of alien introgressions on chromosomes 2A, 3D, and 2B. And I showed you some examples with large structural event, this 5B, 7B translocation. So I'd like to end by acknowledging all the hard people that did the work. This was truly a group effort. A lot of people put a lot of sweat and tears into the project, and to make it a success from a lot of different organizations from around the world. I'd like to specifically acknowledge Cecile Monat and Martin Masher for their analysis of the high C, for structural variants and developing the pseudomolecule assemblies. For the gene and TE annotations, Manuel Spengel, George Haber, and Heidrun Gundelach. For the NLRs, Simon Kratinger and Burkhard Stuernagel. For the Angela elements and identification of the alien introgressions using that approach, Thomas Wicker. For the 2N analysis, Jesse Poland and Liang Gao. For the 5B, 7B translocation, Marcus Kolodzig and Beat Keller. For the visualization tools, Venkat Bandy and Jorge Siri for the Synteny viewers, the linear and the circular plots. And then Cristobal Ue's team, Jemima Brunton and Ricardo Ramirez Gonzalez for that haplotype lure. And also like to thank Curtis Posniak for helping coordinate some of the efforts in the project. And with that, I'll be happy to answer any questions that you might have. And I have the links here for you to copy down for the main website, the data download, the BLAST database, the different viewers, and where we're going to be hosting the seed. So thank you.
Great. Thank you very much, Sean, for an excellent overview of a really exciting project and a great contribution to the wheat community. We do have quite a few questions. Some you've already answered, such as where people can request the seeds. So that is easily obtainable. And in fact, what we will try to do, even though we're not going to be able to have the whole presentation or the YouTube video available until the data is actually published, we will try to put a link to this last slide, in fact, on our website, so everybody can actually see where to get the data. So let's jump into some of the questions. One, is the data freely available for use? In other words, if you download it, can you use it for anything at this point in time? So is it freely accessible?
So if you go to the data download link, which is the third one down here, the IPK one, you're going to have to read that agreement for the use of the data. So right now, it is available for download, but you need to agree that you're not going to publish on this data until we publish our data on this first. Similar to what was done previously with IWGSC RefSeq. So there's a little disclaimer there when you go to download the data.
Yeah, so about a year and a half before we published RefSeq, people could actually start using it and download it and put it onto different platforms. Can people do that at this point? That's also one of the questions. So in other words, can they take this data and add it to their platform and then make it accessible to other people? Or is that also restricted at this point?
Yeah, so I don't have the disclaimer in front of me, so I suggest that you just read that disclaimer. And if you want permission to do something that's outside of what's outlined in the disclaimer, you can send me an email and then I can follow up.
All right.
We are committed to trying to make this data as available as possible to people because we want people to be able to use it. At the same time, we want to be able to respect the hard work that went into generating it. So if you have a specific situation and you would like to get some clarification, the best thing to do is to just send us an email and then we can chat about it some more and we'll see what we can do.
All right, great. So along those lines, one of the questions, and I think you kind of answered this, regarding the annotation data, are they available?
Yeah, so when you go to the IPK download, you agree to that disclaimer, and then you get access to the genome pseudomolecules as well as the gene projections and the TE annotations.
Okay, great. So now to get into some of the more technical questions that have come in, what are you, and if you can answer this, one of the questions is, what's the difference between a land race and a wild relative?
We sequenced as part of this project spelt, which would probably be more outside of what a land race would be. And we see that it's fairly synthetic to the other pseudomolecule assemblies, but there are a lot of unique regions within those. The disadvantage, of course, with the land races is that they're in many cases much more difficult to use in applied breeding than some of the lines that are developed in these advanced elite lines in the global breeding programs. Once some sequences become available for land races, I think it'll be interesting to look at those as well. But For the focus of this project, we really wanted to get at that diversity within the cultivated lines. And as I showed you in the talk, we're seeing a lot of really interesting things, including sequences from wild species that are more distant than even the land races.
Right. Great. Thank you very much for that. So another question, you mentioned a couple of the translocations, which are always really interesting when we're looking at these particular kinds of genomes. Did you check the 1B, 1R translocation by any chance?
I think we did look at that at one point. It's a bit of a challenge because, as many of you have known with just the release of the Chinese Spring genome, it opens this whole new world, and there's so many different things that you could look at with this. I think at some point I actually did look at that. I can't remember exactly what it showed, but the genomes are available for download. So if you're finding neat things in these genomes, by all means, send me an email. We're, happy to work together on these things and to see what you guys are finding as well. So
to follow on with that, do you know what happens at the exact place of the translocation? So are you seeing more transposable elements, less, more repetitive sequence?
Yeah, so for example, for the 5B, 7B translocation, there's this really, really nasty repetitive element right at the boundary at the break point. But we've been able to span it using 5B and 7B specific primers and be able to look at that nasty repeat that's between the translocation event. So yeah, the wheat genome is full of all of these repetitive regions, and they certainly, like has been shown in many other genomes, these repetitive elements play an important role in facilitating some of these events for sure.
All right, great. So another question is, are the ventricosa and excuse me if I pronounce this incorrectly, Timofeevi genomes available on the viewer as well?
Yes.
Okay, great. Are you aware of any other efforts to sequence other crops, non-wheat crops, using the same approach?
I don't really understand the question. So using the same assembly approach, or to look at the introgression analysis specifically about the TVs?
I have an idea. This is a guess, but I think this is in regards to how many, to using the basic platform. So using the NRGene platform, for example.
Oh, yeah. So the NRGene platform is sort of tried and true. It generates really high quality assemblies, and I know NRGene, for example, has made several announcements of different types of genomes, including cannabis and other things. So it's really still a gold standard in terms of genome assembly technologies.
All right, great. Did you see a difference in the number of NLRs between the different lines sequenced?
I think that there was a range, but that those lines are sort of similar in their range. I don't remember the exact numbers off the top of my head. We do have a table of them kicking around somewhere, but there is a difference in the amount between the genomes. But I'd say that they're more or less in the same ballpark.
All right. Will the EMS and the Y-ray mutants be available, and if so, when?
Yeah, that's a good question. So the gamma irradiation mutants and the EMS mutants have been developed, and I think the plan is to advance those lines to a stage where many of those mutations will be fixed, because gamma introduces double-strand breaks and deletions. So they'll be hemizygous initially, and then we need to sort of advance them far enough in order to get them to be fixed. I believe the plan eventually is to get these to be released, but these are still, a work in progress, because they need to be advanced to a stage where they're more usable, and then we can gather more information about those in more advanced lines.
Great. Did the annotation include RNA-seq data?
So We have generated RNA-seq data and iso-seq data, but to generate consolidated annotations between each of the genomes is an ongoing effort. It's a little bit more complicated than it might seem due to just needing to make sure that those genes are annotated identically between each of the genomes. So it is also a work in progress, but it is something that we want to make sure that we get right, and it's going to take some effort to be able to do that.
All right. Another question is, will you make the scaffold order in the pseudomolecules available at some point?
So I'm just recalling what's available on the download site. I think right now they do not have the scaffold placements in the pseudomolecules available. What I could do is I'll see if we can add that to that data download repository, so that it could be available to those who might be interested. But I don't think it's available currently. There's always all sorts of different data that gets generated around these assemblies, and it's we want to make sure that we're providing the data in a sort of easy to use way, and but we could look to see about adding that as well. I'll look into that.
All right.
But I don't think it is available right now.
All right. Did you encounter regions of heterozygosity in the sequences? And if so, how did you deal with those?
So many of these lines are, especially since they're elite lines, they're highly advanced. So most of the loci across all the chromosomes were homozygous. With that in mind, some of them, for example, were derived from crosses, and there was still some residual heterozygosity in there, and that those regions we were still able to assemble them. So there was some, but I would say it was very minimal, since these lines especially were elite lines that were advanced.
All right. The last question, are you seeing highly active repetitive elements between parents and progeny?
Yeah, that's a good question. I don't know the answer to that. I know that some of the TE elements that we did annotate still appear to be the full length and are likely maybe active. When we did the subgenome identification of the TEs, we'd expect that those would be maybe a little bit more static or not active, but we do see some elements that are shared across the subgenomes, which would indicate that they might be active. And at this stage, we haven't looked too deeply, I don't think, at any of the progeny. Between crosses of these individuals, like I said, we developed the NAM population, but that's similar to the EMS. We're working on advancing that.
Great. Sean, again, thank you very much for a really interesting webinar. And just to remind everyone, for this webinar, we are delaying the release of the recorded video until the paper is published at the request of the numerous authors who've done a lot of work on this. And we will also be delaying the release of the PowerPoint slides that Sean has used today. I will say that we will put at least the links to a number of the resources that you can get. We can go ahead and put those out, and we will provide those to you when we send out the post webinar results. So thank you again, everyone, for your participation. And Sean, great presentation. And if you have any suggestions regarding future webinars that you'd like to see, please let us know, because we're always looking for trying to present some of the greatest work that's going on out there. So thank you again, and we'll see you next time on the IWGSC webinar series. Thank you.